Hello. In this presentation, we will be learning about the different facets of an infectious disease process. My dear friends, for any infection to get established, there is an interplay of three primary factors which is collectively called as the epidemiological triad. This triad consists of the components such as agent, host, and the environment. Human civilization has witnessed many such infectious disease outbreaks due to the interplay of these three different factors. Let us now understand the various definitions related to this infectious disease processes. Infection is basically defined as a process where a microorganism enters, establishes itself, multiplies, and invades the normal anatomical barrier of the host, resulting in disease. When the host experiences certain symptoms or clinical features due to this infectious process, it is referred to as an infectious disease. Microorganisms have always shared an intricate relationship with the host. Based on the relationship between the host and the bacteria, we can classify them as parasites, which are primarily those microorganisms that are intended to cause infections. Among the parasites, we have pathogens which causes the harmful or deleterious effects, and then we have another set of microbes called as commensals, which form a part of the normal microbial flora of the host. During situations when the immunity of the host is lowered, these commensals behave as pathogens. That is, these commensals start producing the infectious disease processes. This kind of arrangement or this kind of relationship is called as a facultative pathogens. That is why commensals are also called as facultative pathogens. Then we have another group of organisms called saprophytes. These saprophytes are ubiquitously or universally present in the surrounding environment of the host, like air, water, soil, and organic matter. These saprophytes also causes certain infections whenever the immune system of the host is compromised. So, the point that I would like to make at this juncture is that we should remember that even though commensals are normal, harmless microbial flora and saprophytes are normal, harmless environmental flora, both of them are capable of causing infections whenever the immune system of the host or the individual decreases. Now, the source of microorganisms that are directly linked with the production of the infectious disease process are two in number. These microorganisms can either arise from within the host when they are called as endogenous, or these microorganisms can arise from outside the host when they are called as exogenous source. Here you have to differentiate between two very important terms. One is called as the colonization and the other one is called as infection. To put it in very simple terms, let me just tell you that colonization is a situation where bacteria are present but the symptoms are not seen, or the clinical features of the infection is not seen. The colonization usually occurs from the patient's normal microbial flora and also from the environment. Once the organism gets colonized in the wounds, mainly in the GI tract, skin, surgical wound, nasopharynx, or urinary tract, or for that matter, in any of these, sites of the human body, they remain as innocent bystanders as long as the immune system of the host is compromised, or as long as the host is administered certain antibiotics, usually irrationally, or without even a proper scientific rationale. So under such situations, when the immunity of the host comes down, or when the antibiotics are administered irrationally, these colonizers become pathogens and cause a disease. So infection is that a situation where bacteria is present and the disease is present, and colonization is a situation where bacteria is present but the disease is absent. Now let us find out what are the different sources of infection. Of course, in the top of the list, always it is us. Humans are considered as a very important source of infection. Now, among the humans, you have patients and carriers. Patients are those individuals in a population who are identified to have a particular infection, whereas carriers are those individuals in the population who harbor the infectious agent without showing any clinical symptoms. Such individuals usually shed the organism from the body via contact, via air, or via the droplets. Such individuals are called as carriers. Carriers are usually seen in the infectious disease processes due to inadequate treatment or inadequate immune response. Both these factors result in the incomplete elimination of the organisms from the body. Though they are less infectious than the patients, the carriers are usually more dangerous and are more responsible for spreading the infection in the society. Usually carriers go undetected and they can transmit the infection for a long period of time. Depending on the various individuals, various types of carriers, you can have incubatory carriers who shed the organism during the incubation period of the disease. That is the period between the entry of the organism and the manifestation of the clinical symptoms. You also have healthy carriers who develop an infection without suffering from the symptoms of the disease. Then you have convalescent carrier, those people who have recovered from that particular disease, but they continue to harbor and they continue to shed the pathogen from the body. Depending on the duration of the carriage, you have temporary carriers who shed the organisms for less than 6 months. Chronic carriers shed the organism for indefinite period. Usually the incubatory, healthy and convalescent carriers are temporary carriers, whereas those individuals with certain infections like hepatitis B typhoid, malaria, etc., can become a chronic carrier. Depending on the source of the infection, a carrier can be a contact carrier, that is, a person who acquires the pathogen from a patient, or a paradoxical carrier, that is, a carrier who acquires the pathogen from another carrier. Animals are also considered as a vital source of infection. When an individual acquires an infection from the animals, these infectious diseases are called as zoonosis. The animal actually harbors the infectious agent and acts as a storehouse or a reservoir for the infectious disease process. Then you have different vectors. Then you have certain insects which are both biological and mechanical vectors in an infectious disease process. When the microorganism is just carried from one place to another by the insect, it is called as a biological agent of transmission. For example, when a housefly carries the microorganisms from the sewage and goes and sits on the food which is improperly closed, in such a situation, the microorganism gets transferred from the sewage to the food particle, and the insect, the housefly, is called as the mechanical vector, whereas a biological vector is a situation where the microorganisms undergoes a part of the life cycle within the insect. A classical example is malaria. We say the female Anopheles mosquito is a biological vector for malaria. That means part of the life cycle of the parasite causing malaria, Plasmodium, is taking place within the insect, within this vector, and this mosquito, once that life cycle is over, it transmits that infectious agent by biting the human beings. Then we have soil, which is another very important agent of transmission of infections. Water, especially the contaminated water, and the water which is usually contaminated with the fecal soil, is usually acting as a source of infection. Therefore, it is very, very important to secure the pipes that are meant for transport of the water, both the drinking water and the sewage water. The pipes should be very clearly engineered, and both these pipes should be separate, and there should not be any leakage of the pipes. When such leakage occurs, usually fecal contamination of the water occurs, and we are more prone to such fecal-oral infections like typhoid, for example. And then, of course, food, especially the improperly cooked food, or the refrigerated food, or the food which has been inadequately prepared because of the inadequate washing of the vegetables. All these factors also contribute to the infection. So, to summarize, there are different sources of bacteria ranging from humans, animals, insects, soil, and water and food. Now, an infectious disease gets transmitted through different ways. Transmission of an infectious agent by means of contact usually occurs when a person either directly transfers the agent via the hands. In this situation, it is called as transmission by direct contact. If the agent gets transmitted through inanimate objects called as fomites, it is called as an indirect contact transmission. The inanimate objects can be anything from a table, chair, bench, towel, doorknobs, light switch, anything. So, just to summarize, in a contact transmission, if the agent directly gets transmitted from the hands of the one person to the other, it is called as direct contact. But if it gets transmitted through an inanimate object called as fomite, that is, this particular infectious person may have touched any of these fomites, and if the healthy person touches the same fomite, the healthy person stand a chance of getting the infection from the infected person. Then the organism can also get transferred by inhalation of droplet nuclei. This is called as the droplet transmission. The organism can also be ingested, as I mentioned previously, through feco-oral route due to the fecal contamination of the water sources. The organism can be planted or inoculated directly into the skin by the bite of the vectors or bite of the mosquitoes, etc. The organism can also be transferred from the mother to the baby, where it is called as congenital, and the organism can also be introduced as a result of certain therapeutic, diagnostic and treatment procedures. In such a situation, it is called as an iatrogenic transmission. Please understand that sometimes more than one of these modes of transmission play a very important part in the infectious disease process. So in this particular table, you can see the various infections that are transmitted through these methods. For example, by inhalation, you can get hepatitis B, HIV infection. By inoculation, you can get hepatitis B and HIV infection through the contaminated needles and syringes. By inhalation, you can get measles, mumps, influenza and meningococcal meningitis. By ingestion, you can get food poisoning, salmonella infection. By physical contact, you can get sexually transmitted infections and pneumonia. And from mother to child, you can get certain group of infections which we collectively called as torch infections: toxoplasma, rubella, cytomegalovirus, and the other infections. So, as I mentioned, in an infectious process, there is both lodgement and there is multiplication of the parasite. If that infection occurs for the first time in the host, it is called as primary infection. If the same primary infection occurs once again in the host following recovery, it is called as reinfection. If when the host is having a primary infection due to one organism, he gets infected by another organism on top of this, it is called as a secondary infection. Classical example is the presence of superadded infection like tuberculosis, which is usually seen in HIV infected individuals. So here HIV is the primary organism causing the infection, and tuberculosis is the secondary infection which occurs in the HIV infected individuals due to the low CD4 counts. If the infection usually infects one particular target site of or one particular organ or one particular tissue system, it is called as the focal infection. If the infection spreads from one person to other, it is called as cross infection. And when this cross infection occurs in the hospital environment, it is called as the nosocomial infection. When an infection is acquired as a result of certain diagnostic and therapeutic procedures, it is called as a iatrogenic infection. So this is an example of certain iatrogenic infections. You can see here you have urinary tract infection, respiratory tract infection, okay, you have bacteremia and surgical site infections. So these are few of the examples. Now, what are the clinical effects of these microbes in the host? If that particular infection does not cause any overtly expressible symptoms, then it is called as inapparent infection. Okay, this particular organism does not cause any expressible symptoms, it is called as inapparent infection. If this particular organism enters the host and causes symptoms that are usually not expected of this organism, it is called as a atypical infection. For example, there is an entity called as atypical pneumonia caused by certain organisms like Legionella. In this particular infection, the person will not have any any symptoms as such. So whenever a person has pneumonia, he is supposed to have some respiratory symptoms. So in this particular situation, the person will generally not have any respiratory symptoms. He will have just a mild constitutional symptom for which he will go to the doctor, and the extent of the infection becomes evident only when the doctor takes a chest X-ray. So this particular type of an infectious disease, or this type of an infection where the typical symptoms of a particular infection are not seen, is called as a atypical pneumonia. Atypical infection. And atypical pneumonia is also sometimes referred to as walking pneumonia. And then there is another entity where the infectious agent enters the host, remains in the host, in certain specific regions, and then gets reactivated during the favorable circumstances. And such infections are called as the latent infections. Mycobacterium tuberculosis is one infectious agent that causes tuberculosis, and this particular infectious process sometimes becomes a latent infection, and whenever the time is favorable or whenever the conditions are favorable, this infection gets reactivated and causes the symptoms of the disease. So here you have to understand two different terminologies. Whenever we refer to an infectious disease process, we say that two terms, pathogenicity and virulence. When we say pathogenicity, we are referring to microbial species, that or the ability of the agent to cause the infection. When we say virulence, we are referring to the strain of the microorganism within the same species. Okay, virulence can be compared to the weapons which a soldier has to fight a war, and pathogenicity can be compared to the soldier itself, who is capable of fighting a battle. Now there are two components of virulence. If the strain, okay, becomes more expressive in terms of these weapons or these virulence traits, it is called as exaltation. Whereas if these virulence traits are suppressed, it is called as attenuation. Okay. Usually, when we prepare vaccines, we refer to the term live attenuated vaccines. So, in these kind of organisms, that agent or the infectious agent has lost the ability to cause the disease, but it has retained the ability to form the antibodies. Okay, understand? The pathogenicity is lost, but antigenicity is not lost. So let us now view pathogenesis from the microbial point of view. There are several weapons, as I mentioned earlier, which are expressed by the microorganisms to cause an infection. First, for causing an infection, the microorganism needs to bind to a specific receptor in the host cell. This process is called as adhesion. Once adhesion takes place, the microorganism invades the host cells. Following invasion, it secretes certain toxins. The toxins are secreted from within the bacterial cell, or they are expressed outside. Depending on that, it is called as, or depending on the nature of secretion of the toxin, we say it as endotoxin or exotoxin. Then there are certain extra chromosomal genetic elements which are responsible for the infectiousness of the microorganism. These extra chromosomal genetic elements are called as plasmids. The plasmids do not directly take place in the replication of the organism, but they are definitely present in the organism, and they are capable of getting transferred from between the two same species or between one species to another species. Then we have certain viruses called as bacteriophages, which can harbor these virulence genes, and these genes can be transmitted from one bacteria to another. A classical example is Corynebacterium diphtheriae, which causes the disease diphtheria because of the presence of a tox gene that is carried by the bacteriophage. The Corynebacterium diphtheria by itself does not cause the infection unless and until it has got this tox gene that is present in it. Then we have the communicability of the organism, the extent to which the organism causes the infection by spreading from one host to the another. Certain basic differences here I would like to highlight between endotoxins and exotoxins. Endotoxins are lipopolysaccharides and exotoxin are protein in nature. Endotoxins form a part of the cell wall and does not diffuse into the surrounding. Usually it is obtained only by lysis of the cell. It doesn't have any specific enzymatic action, nor does it have any tissue affinity or a pharmacological effect. It is very, very weakly antigenic, and the antibody neutralization is ineffective. In contrast, exotoxins actions are specifically neutralized by the antibody. It has an ability to produce high amount of antibodies, and the action of an endotoxin is very, very specific in terms of its tissue affinity, in terms of enzymatic action and pharmacological effect. And endotoxins are heat labile, means they are easily destroyed by the heat, and they are actually actively secreted by the cells into the surrounding medium. Usually exotoxins are obtained by the physical means like filtration. So, my friends, this is a nutshell of the pathogenic processes. The organism binds, replicates, releases these virulence factors, okay, and causes an infection. So, once the virulence factors are released, you have two effects. It can directly cause a tissue injury, or it can indirectly activate the host defense mechanisms. They can modulate the immune response of the host and release a lot of these chemical signaling molecules called cytokines. Once these cytokines are released, sometimes they can be detrimental to the host itself. So, to eliminate these organisms, we require antibiotics, and once the antibiotics are used for the right indication, in the right patient, in the right dose, in the right time, then you have a situation where we can eliminate the bacteria altogether. There are certain bacterial products like enzymes such as coagulase, fibrinolysin, and hyaluronidase, which will help in disrupting the intercellular junctions of the host tissue and spread the infection. Similarly, there are certain bacterial appendages like capsule and surface antigen, which are primarily responsible for the infectious disease process. Capsule is also antiphagocytic, that is, it can resist the phagocytosis by macrophages. These are the few ways, or these are the few virulence factors that are involved in the different stages of an infectious process. So, for attachment, you require adhesins, exopolysaccharides, lipoteichoic acid, outer membrane proteins and vesicles. For invasion, you require enzymes and flagella for locomotion. Okay, for surviving inside the host, it requires capsule, the enzymes like IgA, GMC3 and C5 proteases. For producing direct damage, it again requires these enzymes and metabolic byproducts. And for producing indirect damage, there are other various components that are involved. So, another two very important terms that we need to understand related to an infectious process is the infecting dose and the route of infection. The infecting dose is sometimes expressed as the minimum infective dose or a minimum lethal dose. It is in short also expressed as ID50 or LD50. This means that it is the dose of the organism or the concentration of the organism that is responsible for infecting 50% of the individuals. The route of infection is usually dictated by the mode of the tissue damage by the bacteria and the affinity of the organism, it can be any organism, to the tissues. So, when we say infectious diseases, we can broadly classify them into a localized infectious disease and a generalized infectious disease. When we say localized, it can be either superficial or deep-seated. When it is generalized, usually we refer to those infectious disease processes which have entered in one particular site and they have spread from the blood vessels and through the lymphatics and causes these symptoms. Another important term that is usually associated with the infectious disease is bacteremia, septicemia and pyemia. Please understand the difference very carefully. When the organism circulates in the blood, when the bacteria circulates in the blood, it is called as bacteremia. When this circulating bacteria forms toxic products, it is called as septicemia. And when these toxic products produce multiple abscesses or multiple clinical manifestations. It is called as pyemia. So if you look at the infectious disease processes as a whole, we refer to three more terms: endemic, epidemic, and pandemic infection. When we say an infection occurs at a much higher rate than usual, or when you expect an infection to be present at a certain normal rate, okay, our usual rate in a particular geographical area, restricted to a particular geographical area, it is called as endemic infection. For example, in certain places of Andhra Pradesh, fluorosis is one particular disease which infection which is endemic. Okay, or in certain shrub areas you can expect scrub typhus, so that is endemic. If the infection occurs at a much higher rate than usual in a particular geographical area, it is called as an epidemic disease. Usually, this affects a large number of people within a community, population, or region. If it is not quickly contained, this infection spreads to large areas of the world, and when this occurs, it is called as pandemic. Okay. Example of epidemic is SARS epidemic in China in 2003. Example of pandemic is, of course, COVID-19 pandemic in 2020, affecting more than 200 countries. So this is the chain of infection. An infectious agent utilizes certain reservoirs. The reservoir can be people, environment, or water. From these reservoirs, it exits through the excretions, secretions, skin, or droplets, and gets transmitted from one individual to another through contact, droplets, ingestion, or airborne. Once the agent gets transmitted, it enters the susceptible host through the mucous membrane, respiratory tract, GI tract, or skin, and the susceptible host, if the host is immunocompromised due to diabetes, cardiopulmonary infections, immunosuppression, burns, or surgery, then this particular infection establishes itself in the susceptible host, and this vicious cycle keeps on repeating. If you take any infectious disease for that matter, this chain of infection always exists for that particular disease process. Therefore, if you have to break this chain, you need to identify the organisms causing that particular infectious process. You need to identify the mechanisms by which that organism spreads from one person to another, and effectively take measures to control this transmission. And also, you need to identify the hosts who are likely to get infected from that infectious disease process, and we should take active steps to ensure that such hosts do not become the reservoirs of infection in the community. So an infection, to conclude, passes through different stages. Initially, you have the incubation period. Here, the organism enters, but there is no sign or symptoms. Once this infectious period has crossed, the person goes into a prodromal phase. In this particular phase, the person experiences minor constitutional symptoms like fever, headache, myalgia, etc. Then you have the full-blown infection, where the features related to that particular disease is manifest. Then we generally treat the infection during this stage, and once we treat it, the signs and symptoms gradually disappear, and the patient recovers. This is called as convalescence phase. Thank you so much for your patient listening.